Hello everybody, and thanks for the opportunity to present today. My name's Jess Strickland and I'll be talking about convergent oceanographic slicks as habitat for jellyfish. I'd like to acknowledge my collaborators Kylie Pitt from Griffith University and Mike Kingsford and Dean Jerry from James Cook University in Australia, as well as the Minderoo Foundation, which sponsors my project. So our work is based at the Ningaloo Reef in Western Australia. The Ningaloo is the world's largest fringing reef. It stretches 260 kilometres along Australia's west coast, and it's got very, very high biodiversity of, in particular, megafauna. So things like humpback whales, manta rays, and lots of shark species are common at Ningaloo. It's also a tourism hotspot, which is primarily driven by whale sharks. So whale sharks aggregate in huge numbers to feed in the Ningaloo region, and this attracts over 40,000 visitors who want to come and swim with them. But unfortunately for many of these visitors, the Ningaloo Reef is also home to dangerous Irukandji jellyfish species, which sends many people to hospital each year. So for those not so familiar with this group of jellies, Irukandji, the term describes 16 or so species of cubozoan jellies that cause Irukandji syndrome. And while the symptoms do vary from species to species, if you're stung by an Irukandji jellyfish, you're likely to need to go to hospital. You're probably going to be in quite a lot of pain, and we do know that they can actually be fatal as well. So there are two species of Irukandji jellyfish known from the Ningaloo region. The first one is Malobella, which is the small one on the left. It's a classic sort of Charybdid jellyfish. It has a small bell about the size of the tip of your thumb and 4 retractable tentacles, which have the bands of nematocysts that deliver its venom. The other species is Keysingia gigas. It's actually a giant Irukandji jellyfish. It's a big sort of rocket ship shape, and it can get to over 40 centimetres, and it's covered in nematocyst warts. We know very little about both of these jellyfish species. They were described in 2014, but no work has been done on them. All we know is that each year in Exmouth, the tiny town on the doorstep of the Ningaloo Reef that only has a few thousand people, we end up with lots of people going to hospital from Irukandji envenomation. So they can be quite problematic. It's not just Irukandjis that we get at Ningaloo, we get many other species of jellies as well. So we get Cyanea, Crambione, Aurelia, and Pelagia species. That's just the scyphozoans. And in recent years, we've had huge blooms of Crambione mastigiphora washing up and making beaches and surf spots undesirable, unsuitable. So they can be problematic occasionally. One thing that we had noticed when we were seeing jellyfish in any sort of significant numbers is that we were often seeing them in slicks. So we wanted to look at this and investigate slicks as potential habitat for jellyfishes at Ningaloo Reef. In particular, we wanted to answer the question, are there more jellyfish found inside slicks than outside slicks? And we wanted to look at scyphozoans like the Cyanea and the Crambiones it's eating, but also look at other jellies like ctenophores and salps as well. And in particular, we were really interested in looking at Irukandjis like the Malobella shown on the right. Just a quick side note on what is an ocean slick. So oceanographic slicks are these sort of convergent oceanographic features that form these meandering lines of smooth water on the ocean surface. They're a very common coastal feature. You've probably noticed them in your own work. They're formed by a variety of ocean processes such as tidal or headland fronts, eddies or internal waves, and these processes cause an accumulation of organic matter that sort of flattens the surface of water of the ocean. And what we find is these areas have very, very high biodiversity, including zooplankton, so jellyfish prey. So how did we sample them? We wanted to use a combination of 2 methods to sample the slicks. We used video transects, and this was to quantify multiple taxa that were visible on the transects. So here you can sort of see me swimming along in a slick. I've got a crab riding on my shoulder there that you might be able to see. And this was mainly to target the sort of larger, easily visible and easily identifiable jellyfish like the Crambione here. But for smaller, sort of more difficult to see jellyfish like the Malobella on the right, we wanted to use environmental DNA, or eDNA, which is essentially DNA that has been shed into the environment by an organism, and we can actually collect this DNA and use it to study species without the need to physically locate or collect the target animal. So it's quite handy for our purposes. So here you've got photos. I'm scooping up some water, we're filtering it in the field, and then I'm hoping to collect some Irukandji eDNA in that sample, and we take it back to the lab, and we use a PCR assay that we actually developed as part of this project to specifically detect Malobella DNA in the water sample, and that's when we can know whether they were present or not. So to actually sample the slicks, we would go out during the jellyfish season and on low wind days, because that's usually when the slicks form. We would swim 3 video transects up through, see if that works? No, not really. 3 video transects up through the slick, and then 3 outside on alternating sides of the slick. And then we would quantify the different jellyfish that we'd see in those video transects, including into different taxonomical groups. In addition to the video transects, we also used our new Malobella PCR assay to detect Malobella irukandjis in the water samples that we took. So we took 3 water samples inside slicks and 3 out, and for each slick we did 6 PCR replicates, which resulted in 18 PCR replicates inside and 18 outside. And we looked at the rates of positive detection across these PCR replicates as an indication for whether Malobella were present in those samples. Really important to note that eDNA, as many of you are probably aware, it's great for detecting presence of a species, but it's not very reliable for detecting quantities. So we weren't really using it to quantify, more just to detect presence absence. So here's a video of a slick. You can see an Irukandji there. I've circled them. You can see it's really easy to spot the Crambiones, beautiful big red jellies, very easy to count. It wasn't so easy to count the ctenophores, I can tell you that. But the Irukandjis are quite hard to see on those video transects, and this was quite a clear day with good visibility. So this further supported our decision to use eDNA to detect these species in addition to the video transects. So you can see this is inside a slick. Sometimes we did get quite large numbers of jellies, but were there more inside than outside? Well, yes, there were. So if we look here, we're looking at slicks on the x-axis, A to L, and then we've got the number of jellyfish per transect on the y-axis. And you'll notice that for the number of jellyfish inside of transects, which are in the blue, there were significantly more than the jellyfish outside of transects, which are the green. And you'll notice that if we work our way across the top, for slick A, we got 1,500 times more inside than outside. B is 860 times more, 25 times more. The difference gets a bit less, but there are still more inside than outside. And then I, J, K, and L, it was no significant difference, noting that those slicks had quite low numbers. The y-scale is, the y-axis is logarithmic. You'll also notice there's quite a lot of variability, which we're used to being jellyfish scientists. Some slicks had just a few jellyfish in each transect, and others had up to 10,000 or more. So that was the total jellyfish, including across all taxa, but now let's just take a look at a few key taxonomical groups. So Crambiones, you'll notice that we did get significantly more Crambiones inside slicks and outside slicks, but you'll also notice that we did get numbers in 2021 and then even more in 2022. This was the sort of years where we were seeing them washing up on beaches and becoming slightly problematic. But you'll notice that we didn't actually get them in 2023. They just didn't show up that year. Conversely, Pelagia, we didn't get in 21 and 22, but we did get them showing up in 2023, and this was the same for a few other taxa that appeared in much larger numbers in 2023 and we hadn't seen in previous years, and again, significantly more inside the slicks than outside of the slicks. But let's take a look at the Irukandjis, which are what we were really interested in. So there were more Irukandji detections inside versus outside, but please note that over 3 years we had very low detections on the video transects. Only 19 were filmed inside and 8 outside, and there was no significant difference between inside and outside. So that was the video detections. Let's take a look at the PCR detections using the eDNA results. Again, more inside than outside. I will mention this is just a subset of 5 slicks that have done the results for so far, but again, it's not significant, okay? So if we break that up and look at these 5 slicks individually, we've got the slicks on the x-axis and the positive PCR detections on the y-axis. You'll notice that all slicks had positive detection both inside and outside the slicks, except for slick B, which had high detection inside but no detection outside. Now let's compare this to what we were seeing on the video transects. So here we've got the same thing except just for the video transects. And if we look at that same slick, slick B, we had 7 Irukandji seen inside the slick, only 1 outside. If we look at slick A though, we had detections, PCR detections, but we didn't actually see any on the video transects, which was interesting. And this was the same for slick D. We got PCR detections, but none on the video transects. Slick C though, we had very high detections, the highest that we got on the video transects, but quite low detections for the PCR detection, so that was interesting. So what does all this mean? So the first thing to note is that, sorry, I got a bit sad. So what does all this mean? So firstly, we did find that eDNA is a really useful tool for detecting Irukandji jellyfish. There were instances where we found it to be quite sensitive and we were getting positive PCR detections where we weren't actually seeing them on the video transects, but it also sort of highlights some of the limitations of eDNA as a tool. We need to be aware that when we're looking for eDNA detections, we're picking up the molecules, not the animals. So we need to have, we know how jellyfish act, we're jellyfish scientists, but what we need to do is actually also understand how those molecules are acting and behaving in the environment. So understanding the rates at which DNA is shed, how long it persists in the environment, and how that is affected by things like sediment, temperature, UV, and microbes as well is really, really important in helping us to interpret our eDNA results. It might be the case that for small scales like this, where we're looking at slicks and we're interested to know whether Irukandjis are using those slicks as habitat, that it might be useful to use eDNA as a method for detecting presence, but it's still really important to couple it with another tool like video transects, where we can use to actually look deeper into some of that and look at how many were actually present as well. So, in conclusion, our hypothesis was supported. We did see significantly more jellyfish inside slicks than outside, indicating that these could be an important pelagic habitat for jellyfishes, but this difference was not significant for Malobella, noting that we did just have quite small numbers of detections of Malobella species. What we don't know, though, is why. Why is this happening? Is it a case that due to the sort of converging nature of ocean slicks, these jellyfish are being sort of passively aggregated there, or is it a case that they might actually be using these as potential food areas, because we do know that slicks have a very high abundance of zooplankton, so they could be a really important sort of feeding area for jellyfish. We did notice that the taxa and the numbers of jellies varied significantly, as we expect in jellyfish science, not only between years and between taxonomical groups, but even on one day you could jump in a slick in one area and be detecting a whole different species and whole different numbers to a slick in another area. Noting this is a really interesting study site. We're sort of working on the edge of the continental shelf on the west coast of Australia, so it's an interesting area. Finally, eDNA was useful at detecting this dangerous Irukandji jellyfish species. We did have instances where we detected it through PCRs where we didn't see it on the video transect, so that's really important when we're trying to provide information, for example, to a community to manage visitor risk in something like a whale shark tourism industry. It's good to have the most sensitive tools available, but as I mentioned, it's really good to continue to couple it with other tools depending on the questions that we're asking and what we're really interested in to get that sort of full picture. Finally, just a little word of warning for anyone that's looking to delve into the eDNA space. It's really important to take care when we're interpreting our eDNA results, because again, we're detecting the molecule, not the animal, so we need to have a better understanding of how that works out. I think I'll wrap it up there, but thank you very much, and I'd like to invite any questions.